Hi, in this guide we are going to explore how to annotate your NGS scFv data. Here I have an example project where I have a couple of data sets for various types of CSV data. The first one is the single chain variable fragment data sequence with a long read technology covering the full length of the scFv fragment. I also have a data set which was sequenced on the Illumina short read, not covering the full length of the fragment, and finally the similar data set but with unique molecular identifiers. So to do the analysis of this data, we are starting with MiXCR CFL element block. So we are adding this block and we're going to settings. Let's start with the long read data. So the first option is the reference type. With a built-in reference, MiXCR will align the data against the built-in reference of various species. We're selecting Homo sapiens. That's the case of this data. And then we are selecting the built-in order of our construct, which may be heavy linker light or light linker heavy. We are selecting heavy linker light. The next option is the nucleotide sequence of the linker. And this is very important. Here, we need to put the exact sequence of the linker that we have. Typically, it's G4S repeated a couple of times, and in this case, I'm putting the linker that I have in my data. The next option is crucial. It's heavy chain tag pattern. And this is actually a very important option which tells MiXCR how to extract the part of the read that contains the heavy chain of the fragment. So I'm specifying the tag pattern using the syntax developed in MiXCR. So this is the beginning of the read one, then follows the actual read one, that the part that will be used for the alignment of the heavy chain. Then I specify the linker sequence, and it's very important to put lowercase letters here because with lowercase letters, MiXCR will use fuzzy matching of the linker sequence, and this is important because sometimes there are some sequencing errors, etc. And then the rest follows. This is a long read data, so we have only read one, and that's it. So we will extract everything before the linker to be used as a heavy chain, to be used for the alignment of the heavy chain. The next option tells MiXCR how to annotate, how to, well, align and build the clonotypes out of the alignments. In case of long read data, we cover the full length of the fragment, so we are selecting from framework 1 to framework for the full length of the heavy chain. Then we need to put the similar options for the light chain, and here for the light chain, this is the beginning of the read one, then follows anything, and then the linker sequence, and then read one part that will be used for the alignment of the light chain. And again, since we have the long read sequence, it fully covers the light chain as well. Optionally, we may specify the hinge region as well. So by clicking the run button and the analysis will start, it will take some time for the analysis. And once it's done, you can click on the individual sample to see the overview of the quality checks. And here, actually, what we see is that the substantial fraction of our reads is not aligned. There are two main reasons. One reason is the absent barcode, meaning that the pattern that we use here was not recognized in the read, meaning that there are too many errors or something like this in our linker sequence, which prevents extracting this from the raw read. And this is fine. This happens for long read data. The quality is not that great. This happens. No hits, meaning that a substantial portion of the reads was not aligned for heavy chain and also for the light chain, actually, the same story, which again may happen with such artificial libraries, and normally you don't expect great quality here. You can go to the logs of the MiXCR runs individually for heavy and for light and explore a more rigorous reports provided by MiXCR. So now we can explore our scFv sequences by adding clonotype browser block. So let's add it. And in the clonotype browser, let's look on our long read data. And here we have the standard columns like number of reads, fraction of reads, that's abundance, the protein sequence of scFv fragment. Let's select like this sample, for example. Let's sort by the number of reads. And then we have it. We have a scFv full length sequence, amino acid sequence. Yeah, it's in frame. Everything is fine. We have the annotation of the heavy and light, of the variable and joining genes for the heavy and light. There are other columns that may explore like we do normally the overlap, which shows all of the sequences, and as we see there are some out of frames and then some sequences which do not have which contain like stop codons for which we can filter, and there is a productive filter allowing us to set and see only the sequences which are productive, etc. But let's return to the alignment. Let's explore the other options that we have here. Imagine that instead of a built-in reference, we want to use like a scaffold, like a backbone for the alignment. Let's say we have an artificial sequence which we use to construct the library. Then we can put the sequence, the full length scFv sequence containing the heavy linker and light as a reference using the full scFv sequence as a parameter here. And we may specify multiple sequences. This will instruct MiXCR to, well, extract, to use this specific sequence as a reference for the alignment. And if we run the analysis, it will also take some time. It will align and annotate our scFv fragments against this specific sequence. And then if we go to the clonotype browser, we will see the same data. Yeah, let's again sorted by the number of reads. Yes, and we have our sequences as well. So now let's switch to the short read data. So if we use a short read data, let's select here the data sets. That's a short read sequence with Illumina. Let's go to the built-in reference for their example, and let's put the parameters which are relevant to this type of the analysis. In the case of this data set, I have a different linker sequence, and now the important part, the tag pattern. So the tag pattern for the paired end sequencing, and short read is paired end. So for the long read, we have only one FASTQ file. For the short read, we have a pair of FASTQ file for each sample. So for the short read sequencing, the pattern is slightly different. So again, we have the start of the read one, then everything which is read one denoted as read one here as R1 here will be used for the alignment of the heavy chain. Then again the linker follows, then anything after the linker, and now this denotes the separator between reads. So everything before this is read one, read one of our FASTQ pair, and everything which is after this symbol is read two, which we just ignore by providing the beginning of read two and anything. This is ignored. Only this part from read one will be used for the alignment of the heavy chain. In this specific case of this library, the read one covers a part of the heavy chain, and in the case of this library, it covers CDR3 to framework 4, and the light chain tag pattern contains both read one, which is at the end of the actual read one from the FASTQ file, right after the linker, and the whole read two, which covers the most part actually of our light chain. And for the light chain, I'm specifying framework 1 to framework 4, meaning that our short read data covers the full length of light chain and only a part of the heavy chain. So with this, I can click Run button, it will start the analysis, and once the analysis is complete, we can go to the clonotype browser to explore our short read data. And what's important here that the short read data here we have the full length of a scFv fragment, and this is one of the key features of this block, because normally you need the full sequence, you need the full sequence, the full protein sequence of the fragment, and if we make visible the nucleotide sequence of our fragment, then you will see what I mean. So this is a nucleotide sequence, and you see the lowercase letters are the letters that were imputed from the germline. They were imputed from the reference. If we use the scaffold sequence, then this part will be imputed from our scaffold sequence. And the uppercase letters, they're actually the part of the scFv which is covered by our library, and this is the part coming from the read one containing the heavy chain, then the linker sequence, and finally the part of our NGS data that covers the light chain. And for the light chain, we see it is fully covered towards the end of the framework 4. So that's it. Now let's go back to the alignment parameters and finally use short read with UMIs. Just for the illustration, how you would modify the patterns to tell MiXCR to also extract the UMI. So basically, what we need to do is just to tell MiXCR where the UMI is exactly located in our sequence and data. So in the case of this data, the UMI is located in the beginning of read one. Just 12 letters of the read one correspond to the unique molecular identifier. Yeah. So I am putting this here to tell MiXCR that this is a UMI, and what is crucial, I also have to specify the UMI the same way in the light chain tag pattern. So in both cases, whether we're extracting heavy or whether we're extracting light, we have the UMI, and this is how we do this. So for the heavy chain, it's the beginning of read one, then the part which contains the heavy chain follows, and then the linker, and then whatever. And for the light chain, we have UMI, then whatever, then the linker, then the part read one and read two, which is used for the alignment of the light chain. So when we do the analysis, it will align and to reconstruct the clonotypes, which in the clonotype browser, if we look now, instead of number of reads, we'll have number of UMIs, and we have our scFv sequences here. So that's it for today. Thank you very much, and stay tuned for other guides.